Hey, welcome back. Now let's look at the white blood cell and red blood cell. So look at their structures and their functions. Okay. Now we've already said the both white blood cell and red blood cell they are made in the bone marrow. They are from same type of stem cells. Now I will encourage we go back and study about the stem cells, so you get to understand what we say they are from same type of stem cells. So we have that. We've done that in our previous class. Okay, so when you talk about stem cells are, will I use the word a dormant cells? They don't have a function. They just wait to pick up a function when the need arises. Okay, so there are stem cells responsible for certain type of cells, while others are responsible for other type of cells. So that's why we say they are from same type of stem cells, which are all produced by the bone marrow. Okay, all right. Let's look at white blood cell. So what is white blood cell? Now when we talk about white blood cell, it's also referred to as leukocytes. So take note of that. Leukocytes, they are made in the bone marrow, like we said before. Their function is to fight diseases. Now this white blood cell, they are of two types. So we have two main types actually. There are more than two though, but these are the two main types. So we have the one we call the phagocytes, and we have the one we call the lymphocytes. So let's take the one after that. Let's start with phagocytes. Now, phagocytes, they are produced throughout life. They have a function of patrolling the blood, the tissues. Remember when we talk about tissue fluid, we talked about the fact that phagocyte can pass through the pores of the capillaries. Okay, so the function, they patrol the bloodstream, tissues, and organs. They remove dead cells and pathogens. Now, this process of removing those dead cells and pathogens by the process we call phagocytosis. That is by engulfing. So they engulf, destroy, and then they engulf and digest. Let me use word digest, not destroy. So you can see the image here. You can see the white blood cell, the phagocytes. You can see the bacteria when they swallow it up. At this point, they swallowed it up here. So when they swallow it up, the bacteria are not digested. There are particles inside the cell that helps in the digestion of this the bacterial cells, and then after that, some of them will die, some will live on. Okay, we'll talk about that later. So if you check chapter 4 of your AS biology, you get to understand the structure of these phagocytes. Where we talk about the cells, so you get to know more about phagocytes. Okay. Now we say it involves in a non-specific defense. Now when I say non-specific, we mean non-self. Now there's what we call self-recognition and non-self recognition. Now that's how this white blood cell works. Now when pathogens or foreign bodies gets into the body, it's entering there as a foreigner, so it is not part of the body. Okay, so once the white blood cell senses that this is not part of the body, they will trigger a response. That's what we meant by non-specific. Now, once they see something that is not part of it, they will send a code called non-self recognition. And once that happens, there's going to be an action will be triggered. Okay, so there are different different antigens, and they have their different. When we talk about sensing of this recognition, if you remember, if you look at the membrane of a cell, there are different proteins there that send signal. These are the signals that they use to understand if this is part of us or not. So I will advise again, go back and study the cell membrane and get to understand it better. Okay. Now, when it comes to appearance, there are lobed nuclei and there are also granular cytoplasm. Now, look at the phagocytes. Now, phagocytes is of two types. Okay, so we have the monocytes and we have the neutrophil. Now, look at their structure. You can see, if you look at the monocyte, you can see the shape. The shape looks loop-like form. Now look at the nuclei. You can see the nuclei. See the shape of the nuclei like a bean shaped. Okay, that's what I mean by looped nuclei. Now, when it comes to granular cytoplasm, they have many vesicles. Now, look at the neutrophil. You can see this is a loop. This is another one. So they are multiple. They are multi-looped. We use the word multi-looped nuclei. Okay, so that's what you use to identify these two. So please take note of these of the structures You can see in the exam, they will ask you to identify the cell. So look at, if they are multi-looped, that means they are neutrophil. If they are looped, one looped nuclei, that means they are monocyte. Now when it comes to lymphocyte is round, and one thing that differentiates in structure wise, one aside from the fact that their nuclear is round, look at the size of the cytoplasm. Now lymphocyte, they have smaller cytoplasm compared to monocyte and neutrophil. So that's another way to identify them. And then also remember, the engulfment is usually done by the phagocytes. Okay. All right, so let's move on. Now, having understood that, so let's move on to the real structure of the neutrophil. Now, example, we have the phagocyte. We're looking at the neutrophil. They will say they are multi-looped. Now, if you look at the structure here, you can see this is a micrograph. Now, micrograph, the ones viewed under the microscope. So this is a neutrophil. You can see the multi-looped nucleus. Now they have receptor protein on its membrane. Remember, receptor protein is what they use to identify. I told you about the proteins I see within their membrane. So called the receptor proteins. They are the ones they use to identify pathogens as non-self. Now, phagocytes can only fight or destroy pathogens if it is being identified as a non-self. Now, if the pathogen identifies itself as self, then they're not going to do anything. They will take it as self. So they can only fight non-self and embrace self. Okay. Now, when there is an infection, large numbers are released from the bone marrow. Now they accumulate at the site of infection. So once there's an infection, all of them gathers towards where that infection is. Okay. Now that is the reason why when there's a cut, after in the healing process, will produce what we call pus. It will produce what we call pus. Now those pus are those white fluid. Think of pimple. Those things you usually remove from your pimple. That's actually the neutrophil. Okay. Now, mind you, they are short-lived as well. They use the word they are suicidal in nature. Anytime they engulf and digest pathogen, they will die. So their lifespan can be within a few hours or days. Okay. So that's why I use the word short-lived. So when they ingest, when they swallow up, when they engulf pathogens, they will digest the pathogens and they themselves will die alongside the pathogen. And when they die, they will form those spores. The common one is one you remove from your pimple. When you press your pimple, that's a typical example of dead neutrophils. Okay. All right. So having known that, so let's move on to monocytes. Now, monocytes. If you look at monocytes, this monocyte, you see that within the blood, but when they mature, they mature to become a macrophage. So mind you, the difference between monocyte and macrophage. Macrophage is a matured monocyte. So monocyte is circulates within the blood, but once they mature, they will move into organs like liver, lungs, spleen, and so on and so forth. So they move into the tissues. But when they're circulating in the blood, they circulate as monocytes. So once they mature, they move into the body organs. Okay. They also have lobed nucleus. Remember, their own is lobed. Their own is not multi-lobed. It's lobed and it's kidney bean shaped. So they are larger than neutrophil in size. So neutrophil is smaller than monocytes. Now they have receptor proteins on its membrane, same for identifying pathogens as itself. Now monocytes, again, I talked about that. They mature into macrophage when it leaves the blood and enters the organ. Okay, I've talked about that. They are long-lived. Now they are not like neutrophils. They are not like neutrophils. They are long-lived. Okay. So only neutrophil they will die after ingesting pathogens, but others wouldn't. Now look at the image, look at the structure, understand the structure. You can see the shape of the cell membrane. Monocyte is smooth while macrophage is rough. So take note of that in case if you are given that in the exam, you are asked to identify them in the exam. So look at their structure. Okay. We talked about the fact that it's produced within the bone marrow. So we'll talk about the lymphocyte now. Now, lymphocyte is involved in specific immune response, same. Now, respond only to a specific non-self antigen. Now, mature lymphocyte circulates in the blood and lymph. Okay, take note of that. Mature lymphocyte circulates in the blood and lymph. Now, they also accumulate at the site of infection. also accumulate there at the site of infection. Now, they are for appearance. You can see the image here. Lymphocytes, they are smaller than phagocytes. Lymphocytes are smaller than phagocytes. They have large round nucleus. You can see the nucleus, large round nucleus, and they have little cytoplasm. Look at the micrograph here. You can see the micrograph. You can see the nucleus. You can see the small cytoplasm remaining. Okay, that's why I say they have little cytoplasm. Okay, so moving on. Moving on again, they have two types. Lymphocytes are of two types. Their types is based on where they mature. Now, bone marrow makes them, but they mature in different places. Now, the first one is B lymphocyte, the one they call B cells. Now, B cells, they mature within the bone marrow. Now, the B there stands for bone. B for bone. Okay, so they're the ones that produces antibody. You get to talk about this antibody in I think in chapter 11. In chapter 11 of the AS. Then we also have the T cells. Also have the T cells. That is, the T stands for thymus. This one matures within the thymus. So that's what brought about the B cells and the T cells. Okay. We'll also see how they function. Now, there's also what called the memory memory T cells. You get to see how memory T cells function. Okay, like I said in chapter 11. Okay, so that's what we have the B cells and the T cells is based on where they are produced. B cells produces antibodies, while T cell doesn't produce antibody. Both of them work together to defend the immune system. So when we get to that point, you get to see their different functions. Okay, so here is an example. Now you can pause the video for a few minutes or a few seconds and try to identify all these cells. Now this is a blood smear. Now when you smell blood on a slide and you view it under the microscope, this is what you will see. That is, remember, it's going to be stained. They have to apply some stains in order to bring out the appearance. Okay. Now this is the samples. Now, if you are given this and you're asked to identify each of the cells here, so which one would you refer to as the neutrophil? Which one will you refer to as the lymphocyte, and which one will refer to as the monocyte? Okay, so pause the video, like I said, pause the video for a few seconds, try to identify them, then after that, play the video and let's see if you're correct. Okay. Now, having done that, so let's look at this now. So this, and this are the lymphocytes. So these are the lymphocytes. Let's go back and see if we're correct. So these are the lymphocytes. So look at the image here. Okay, we are correct. So that's the lymphocyte. You can see the shape here. So that's the lymphocyte. So let's see the next one. So these are lymphocytes. So what about these ones? These ones are easy to identify. These ones are easy to identify. So these ones are the what? They are the neutrophil. So these are the neutrophil. Then this last one. Now remember, neutrophils, they are multi-lobed. You can see the multi-lobed structures of the nucleus. Now look at this one, the last one. These are monocytes. These are monocytes because of they are one lobe and it has a bean-shaped structure. Okay, so we've identified the three of them. So we have the lymphocytes, we have the neutrophil, and we have the monocyte. You might be given these structures in the exam. You'd asked to identify each one of them. Okay, so having achieved that, let's move on. Okay, so the difference, if you are differentiate, remember in A level, there's a way you differentiate. There's a way you differentiate things. Not like this. This one is just to give you the points. So here, differentiating between white blood cells from RBC. So say first point, say white blood cell contains a nucleus. Now if it's in the exam, you're going to draw a table, mention the specific. Let's say here we have the nucleus, and here we have the WBC, and here we have the RBC. So here we say nucleus present, absent. Okay, like that. So that's how you do it in the exam, the format you use in the exam. Next one, we said they are mostly larger than erythrocyte. Remember, erythrocyte is white blood cell. So red blood cell. White blood cell, most white blood cell is larger than red blood cell, except for lymphocytes. Lymphocyte is smaller in size than white blood cell. Okay, so generally they will say white blood cells that is larger than red blood cell, but have it in mind that there's a type of white blood cell that is smaller, which is the lymphocytes. Then there are spherical or irregular shapes for white blood cell, while red blood cell they have what we call the biconcave disc shaped. Okay, they are biconcave disc shaped. Now, another thing, white blood cell, they have a granular cytoplasm that is for phagocytes, while red blood cell they don't. They don't even have. Okay, that's not the nucleus now. White blood cell they have granular cytoplasm that is phagocytes, while red blood cells doesn't have. Okay, so next we'll talk about the red blood cell, also known as erythrocytes. Now, red blood cell they are short-lived. They live for 120 days. They are responsible for transportation of oxygen to the body tissues. Features, they are small and flexible. They have diameter of about 6 to 8 micrometer. They are able to squeeze through capillaries. Now they reduce diffusion distance. Now they are able to fix through the capillaries. Now the reason is when they fit through, when they feed or squeeze through, you can see they are closer to the walls of the capillary. Let's assume that, let's take this to be the capillaries. So you can see that they are closer to the walls of the capillaries, thereby reducing the distance, the diffusion distance. So that's why capillary is very good when it comes to diffusion, because they are thin and they have thin wall, and they're also small lumen. Okay, so they are biconcave disc. This biconcave disc shape is what brought about the increase in surface area. Now, when we talk about biconcave disc, think of it as a sunken center. You can see the inside here. The sunken center. This sunken center is bringing about increasing the surface area. So this is what we meant by biconcave disc. You can see. So this place is a sunken center. Okay, they have no nucleus, no mitochondria, or endoplasmic reticulum. So because they don't have all this, they create more room for hemoglobin. Now they maximize the number of oxygen they carry. Now hemoglobin is not red blood cell. Hemoglobin, hem there is blood, globin there is the globular protein. Is a protein that attracts oxygen, that has a very high affinity for oxygen. Okay, so hemoglobin, talking about hemoglobin, they are globular proteins. You can check that in the previous chapters when we talk about proteins. They are globular proteins. They are found in large quantity in red blood cell. Now they are made up of two alpha globin chains and two beta globin. Now you can see the alpha and the beta. Like I said, you can go back to the structure, the proteins. You get to understand this. Now each of these carries one hem group. That's why they got the name hemoglobin. Hem is there, and then we have the globin, which is the globular structure. Now this structure is called quaternary. Quaternary is because it comprises of all these different chains. Okay, each chain contains one hem group that has ions. So you can see the ion here, and then they bind one molecule of O2, meaning that total of one hemoglobin carries 4 oxygens. One hemoglobin carries 4 molecules of oxygen. That's what it's trying to say, because each hem attracts oxygen. Okay, so it's the hemoglobin that has the high affinity for oxygen. That's why each of them attracts oxygen. Okay, so here we said when hemoglobin binds with oxygen, it forms oxyhemoglobin in the lungs. Oxyhemoglobin. So the binding happens within the lungs. Okay, so this is the formula. So this is hemoglobin plus 4 molecules of oxygen to form the oxyhemoglobin. Okay, so that's for hemoglobin. Like I said, if you want to know more about hemoglobin, you can refer back to the previous chapters. There's a chapter for protein where they talk about protein in general. So I would advise you go back there and then get to understand proteins better. Alright, so we're going to stop here. We will continue again in the next video. See you in the next video. Bye.